Session on mollusk and seaweed health. May I now invite the chairs of the session, Dr. T. Rajaswaminathan and Dr. Kalaya, who will be joining us soon to, so please occupy the dais. May I now also introduce our session coordinators for this session, Dr. Bhuvneshwari and Dr. Vidya Rajendran from SIBA. We have two speakers, two keynote presentations today, Dr. Naoki Ito and Dr. Claire. Dr. Claire's presentation will be online. Yeah, now I hand over to the session coordinators to conduct the program.
Thank you, Madam. Now I will introduce the chairperson, Dr. T. Rajaswaminathan, sir. And Dr. T. Rajaswaminathan is a veterinary microbiologist with 25 years of experience on fish health and currently heading the Microbiology, Fermentation and Biotechnology Division in ICAR Central Institute of Fisheries Technology, Kochi. Dr. Swaminathan specializes in fish cell culture and developed 25 fish cell lines, and he had isolated 5 important emerging finfish viruses from different mass mortalities. And he had been awarded with a National Fellowship by Indian Council of Agricultural Research for developing rapid field diagnostic kits and vaccine for managing goldfish hematopoietic necrosis herpes virus during 2013 to 22. And he had been conferred with Fellowship of Indian Virological Society in recognition of the contribution to aquatic virology during the year 2024. Doctor he has published 150 publications in peer-reviewed journals, and he is doing disease management consultations to ornamental fish farmers across the country, and he is involved in National Surveillance Program of aquatic animal diseases in the country. He is a member in the committee of Shrimp Broodstock Multiplication Center for the establishment of aquatic animal quarantine, and he is the member in the subject expert committee of INSPIRE's Fellowship, and he is an expert member in DST. Thank you.
So very good afternoon to everyone. After the lunch, the hall has become thin now. So we hope that people, they may be joining after maybe some time. So this special session on aquatic and seaweed health, we are having 2 keynote addresses during this session. There is no oral presentation. Dr. Clary, he'll be joining online, I think. He'll be talking about the algal pathogens. Another one important talk we are going to get from Nakoi. So he'll be talking on health management of mollusc diseases. So I am happy to join the podium with Dr. Kalaya. So we heard much from her yesterday on EHP. So thank you very much. Now I welcome Dr. Nakoi to talk on pest management on mollusk diseases. So we know that mollusk, it is important pathogen. So, important food animals in the aquatic animal web. So, if you are if you are talking about mollusk, it is a very maybe bottom feeder. So, it is known for pathogen to be accumulated. So, by keeping the importance in that view, OIE, that World Organization of Animal Health, they listed certain diseases which has been should not present in the any trade. So more than 7 pathogens has been listed by OIE, which is having importance of mollusk pathogen. So it is it is a good time to hear about the management of mollusk diseases from Dr. Nakoi. So the time allotted to him is 30 minutes. Already we are 15 minutes delay. So 25 minutes he will be presenting. Then another 5 minutes he will be open for the discussion. So thank you very much. I hand over my podium to Dr. Nakoi.
So before the starting, may I welcome Dr. Kalaya, Srituniya Lekchana Dangtip. Dr. Kalaya is a leading expert in shrimp health and aquaculture. serving as a principal researcher and head of the aquatic animal health research team at Biotech Nasdaq, Thailand. Recognized globally, including the Newton's Prize Chairman's Award and Stanford's top 2% scientist in fisheries, Dr. Kallaya leads the shrimp immunology platform at Mahidol University and contributes to the international initiatives like International Network for Shrimp Health. Her research focuses on shrimp pathogen interactions, disease prevention, and genetic improvement of black tiger shrimp for growth and resistance. Her latest work explores viral accommodation strategies to develop shrimp vaccines and enhance viral tolerance in breeding programs. Now, may I invite Naomi?
May I now invite our keynote presenter, Dr. Naoki Ito, to take the rise. Dr. Claire, as I mentioned earlier, she'll be joining us online.
So I'm happy to share about the speaker of today's session. So he is currently a full professor at the Graduate School of Agriculture Science and Life Sciences at the University of Tokyo. His specialty is infectious diseases in aquaculture species, with a particular focus on researching viral, bacterial, and parasitic diseases in marine mollusks, such as oysters, clams, mussels, and abalone. So through various approaches, including the epidemiological surveys, identification and elucidation of this pathogen life cycle, histological observation, and the study of the host defense mechanism, he aims to advance both scientific understanding and the potential treatment of the mollusk diseases. So now I welcome him to present his talk. So as I told to you, so half an hour has been allotted to you. You restrict your talk by 25 minutes. We'll be giving a ring at 25 minutes. Then 5 minutes will be open for the discussion.
Okay, thank you very much. Good afternoon, everybody. I'm Naoki Ito from the University of Tokyo. So today, I'm talking about mollusk diseases, but probably most of you are not so familiar of marine mollusk things, because please look at for example, the logo of this conference. We can find crab here, fish and shrimp here. Where is the mollusks? Can you find this one? Much smaller than bacterium. So this is how much little attention is paid to mollusks, right? But actually, it's very important. So I would like to. I'm very happy to share the information today. So, but I think maybe most of you may think me, why this guy is doing such a strange research things, right? But actually, the mollusk disease was not my first choice of studies, but the choice of my supervisor when I was a student. And this guy is Professor Kazuogawa. He was my supervisor at the graduate school. And by the way, this guy is Dr. Kiyokuni Muroga. He's also very famous fish pathologist for fish and bacterial and viral diseases. And because Dr. Ogawa is very famous for fish parasites, so I wanted to study fish parasites. So one day, I visited his laboratory. And I still remember the first day when I visited his laboratory. He asked me, do you prefer papa or oyster? So I expected, okay, he's so kind. He asked me what I want to eat for welcome dinner for me. So I answered, of course, puffer fish. It's very expensive, so I said, puffer fish. But he said, okay, but the actual meaning of his question is, parasite of puffer fish or parasite of oyster? Although I answered puffer fish, but he said, okay, you have to study oyster disease. So I don't know why he asked me, but anyway, he decided my way of the life. And but I'm very happy, appreciate him because based on his decision, I'm here today. So I'm really appreciate him. So okay, so moving to the main talk. So probably you don't know so much about how serious mollusk disease for aquaculture. So this is very famous figure to explain about the impact of infectious diseases on oyster production in France. Probably you may know that, but I would like to explain. In France, originally, 2 species of oysters are cultured: Portuguese oyster and flat oyster. But in 1970s, due to the outbreaks of virus infection, the production amount of Portuguese oyster suddenly declined. And alternatively, French oyster farmers introduced oysters from Japan, the Pacific oyster, and they started the Pacific oyster culture. Later on, 2 protozoan parasitic diseases, Maltaria refringens and Bonamia australia, destroyed the flat oyster production. So the alien Pacific oyster became the major cultural species in France. But in 2008, the outbreaks of Australia herpes virus, and in 2011, I remember, a Vibrio infection caused, again, a big problems for oyster production. So I think now you can understand how the infectious disease have big problems on aquaculture by mollusks. So now I think you understand, but still the researches on marine mollusk disease have much less progress compared to those studies for fish and shrimp diseases. And I think there are many reasons, but I think one of the biggest reasons is the difficulties in diagnosis, identification, and handling of pathogens. As major pathogens of marine mollusks are protozoan parasites, and bacteria, and viruses are known. These are the examples of protozoan parasites of mollusks: Parkinson's, Maltaria, Haprosporidium, Bonamia. But maybe you do not know so many about these things, and maybe taxonomic groups of these protozoan parasites are totally different from the protozoan parasites for fish and shrimps, I think. So for diagnosis or identification of these pathogens, we need expertise techniques and knowledge. And also, except for Parkinson's also and Parkinson's species, life cycle of these pathogens are completely unknown. So it means we cannot do any experimental infection for these important pathogenic groups. So it means it is really difficult to conduct researches for these parasites. And bacteria, actually, Vibrio and Francis, maybe it's a common pathogen for fish species, so maybe it is not so difficult to study pathogen itself, but the problem is, unlike fish, mollusk contains a lot of symbiotic bacteria inside the body. So it is very difficult to isolate these pathogenic bacteria only. Well, also it is also difficult to differentiate pathogenic bacteria from non-pathogenic symbiotic bacteria. And the viruses, Australian herpes virus, virus, and also asper-like virus, which has been recently identified. These viruses cause very serious problems. But no cell line of marine mollusks have been established so far, so we cannot isolate these bacteria using cell line. And also we cannot propagate these viruses in vitro. So there are still many difficulties conducting experiments or researches for these mollusks pathogens. But recently, fortunately, a lot of molecular techniques have been introduced to marine mollusk diseases research. For example, a lot of specific PCRs have been developed for many known pathogens. So now we can do a diagnosis and identification of these pathogens without specialized techniques or knowledge. So it's very useful. And also, as for identification of new pathogens, this is a very good example conducted by Dr. Matsuyama. He identified 2 important marine mollusk pathogens using next-generation sequencing to find unique gene specific in diseased animals. So using that techniques, he identified these 2 pathogens which had never been identified for more than 40 years. So improvement of the identification of novel pathogens have been now accomplished. So now the situation is much better. So I think now mollusk disease surveys enter a new stage focused on practical countermeasures. However, vaccination and antibiotics treatment, which have been commonly used to control fish disease, cannot be applicable for molluscan diseases. As you may know, mollusks lack of adaptive immune system, so vaccination is not possible. And for antibiotics treatment, oral treatment by mixing feed and drugs are not practical, particularly for filter feeder bivalves. So maybe we have to think about something different approaches to control molluscan diseases. Fortunately, there are some successful cases for selective breeding for disease resistance in some bacteria, some diseases of oysters. So I think this approach is compromising, probably, but it is too early to rely on this method, because this method is maybe can be applicable only for mollusks with hatchery techniques and disease with very good experimental infection models. So today I would like to say that how we can approach to another strategy, and I think maybe field surveys can make a good, big contribution to the improvement of biosecurity and also improvement of aquaculture management. So today I would like to introduce some of our experiences contributing to these strategies. So at first I would like to introduce our research contributing to improve biosecurity against ovary enlargement disease of oysters. This disease is characterized with the development of these nodule-like structures in soft bodies of oysters. This is a transverse section of diseased oysters, and these are nodules. And histological observation revealed that these nodules are abnormally developed ovary. And in the ovary, you can see many egg cells, oocytes, having protozoan parasites. These are protozoan parasites. And these protozoan parasites, Malterioides tumens is a name, is a causative agent of this disease. Actually, I like these beautiful oysters, but the consumer does not accept it. So these diseased oysters lose, of course, market values. And also, diseased oyster shows high mortality, maybe because by exhaustion to produce these abnormal developed ovary. So sometimes high mortality, also problems. And the prevalence sometimes reach up to 40%. So this is one of the most serious disease for Japanese oyster culture industry right now. And this disease has been reported in mainly western and southern part of Japan, but not at all in north or east part of Japan. However, in 2021, one day I got a call from the farmers from this area, and he said, "Oh, I found this parasite disease." But suddenly jumped to here. I cannot clearly understand, so I tried to collect more information from him, and he said, this disease is only found from the seed introduced from Miyagi area. And in this area, a local seed is also produced for aquaculture purpose, but this disease was not found in local seed at all. That's the information I got. So Miyagi is here. But I was a bit skeptical about this information because this disease has never been reported in Miyagi area. But anyway, we collected oyster seed produced in Miyagi area by PCR, and as a result, yeah, he was correct. Very high PCR positive ratio in the seed in Miyagi area. In order to confirm that the Miyagi seed only developed disease, we maintained Miyagi seed and local seed in same area and examined them periodically. And we confirmed that only Miyagi seed developed the disease like that, while the local seeds are all fine. So from these results, we think probably the oyster farmers in Miyagi introduced oysters from this endemic area, and then the parasite settled in Miyagi area. And then the parasite was transferred to northern part of Japan, so maybe that's the reason. And Miyagi is one of the largest oyster seed provider across Japan for aquaculture purpose. So I publicized this data public and to raise awareness of biosecurity among oyster farmers in Japan. So I think maybe now the disease has not been spread so far, but but anyway, this is how I contributed to a biosecurity measure from field surveys. Okay, second example is, I would like to explain about our contribution to improve scallop culture management against Franciscellosis of yellow scallops. This disease was first found from the scallop with adductor muscle having some orangish regions. And the histological section shows that this region contains a lot of massive inflammation of the hemocytes. And this inflammation is caused by some intracellular bacteria. And sometimes diseased oyster shows a shell deformity like that. And this very similar disease had been found in Canada, in scallop culture in Canada, and the Canadian researcher identified the causative agent as Francisella harriot cider. Through collaboration with Canadian researchers, we found that this disease was also caused by the same pathogen, Francisella harriot cider. And in the epidemiology survey, we found that the prevalence of this disease was very high in farms with low survival rate, like that. And also later we conducted experimental infection using the isolated bacteria, and you can see the challenged scallops showed much less survival rate compared to control one. Sorry. So we can say that this bacteria causes high mortality of cultured scallops in Japan. And during epidemiology survey, one field collaborator said that, okay, but when we did the density adjustment area, the scallops seem to die less. What? What does it mean? So please just let me explain a little bit about the scallop culture in Japan. In Japan, usually in July, we collected a wild spot using net collectors. And in August, those scallops are stocked initially in these baskets. And in September to October, those scallops seeds are again stocked in other basket to adjust rearing density. And in March to April, those oysters are tied up to those rope using the method called ear hanging and grown until harvest. That is an oyster culture program in Japan. And this means, what does the guy mean is that if we conducted the density adjustment operation conducted a little bit earlier, the scallop in that group shows less mortality That's what he said. So in order to confirm his mentioning, we set up these experimental groups with different initial stocking density, low and high, and also with different timing adjustment, like September for early and October for late. And we examined all the groups at next March. And as a result, the group with high initial stocking density and the group with late layering density adjustment showed very low survival, high prevalence of the bacteria, high ratio of shell deformity, and worse growth. We still do not know how much the association of layering density and the impact of the disease, but a local authority now recommended that low density at initial stocking and early operation for density adjustment as a countermeasure to these bacterial diseases. Okay, so at last, I would like to introduce this. This is just a preliminary survey, but I would like to share this information. So this is research is to improve abalone management against amyotrophy of abalone. In Japan, abalone seed is produced for release to help natural stocking resource recovery. And many hatcheries have suffered with a very serious problem of this. And this disease has been found in 1980s. And the typical symptom of this disease is amyotrophy of foot muscles, shrink of foot muscles. And the mortality is sometimes very high, and sometimes reach to 90% of 0-year juveniles during the first spring season, like April to July in hatcheries. And it has been known that this disease was infectious disease, but the causative agent has not been identified for maybe 40 years. But recently, Matsuyama et al. Found that this disease was caused by a novel virus type, abalone asper-like virus. And also, he developed qPCR to detect this virus, so we used that method to examine this abalone hatchery. And the upper graph shows, in the upper graph, black dots indicate the copy number of the virus, and the lines indicate the cumulative survival rate. And the below graph shows the temperature during the survey period. And as you can see, in the 0-year abalone, drastic decline of the cumulative mortality, survival, and also the high copy numbers are detected in the water temperature ranging from 17 to 25 in 2021, and in 2022, those sudden decline of survival and the high copy number was found around between 14 to 23 degrees Celsius. But on the other hand, in the 1-year-old abalone, even in the same temperature range, sudden decline of survival rate or high virus copy numbers were not observed in the right figures. So from this result, we consider, okay, so maybe the older, larger abalone are resistant to this virus infection. So in order to confirm our hypothesis, we asked our field worker to produce abalone seed a little bit earlier than ordinary days. In ordinary year, usually the abalone seed is produced in November, but we asked them to produce abalone seed a little bit earlier because, how to say, we consider that maybe the abalone produced earlier, maybe they grow much enough before the outbreaks of the disease. So we produce the seeds like that, and we maintain them and examine for a while. And we set up similar experimental groups. And as a result, low mortality and low viral copy of mortality and viral copy numbers are very, very low, even in spring as the disease inducing temperature. But still, this is just preliminary, and we should not exclude some possibility that, for example, these seeds are genetically resistant to disease, and also some unknown environmental conditions suppress the disease outbreaks or suppress the propagation of the virus. So still, it's just just the preliminary results, but I think I'm very still happy to find at least one research direction to control these diseases. Okay, so these are the final remarks. So there is no effective control for molluscan disease by vaccinations and antibiotics. So we have to think about something totally different, and also we need to think about many countermeasures from many approaches. And in that context, I think there are many clues for disease countermeasures in production sites, fields. So I think the field-based surveys, as well as communication with field workers, technicians, and of course farmers, are very, very important to get those clues. And in addition to get some of those clues, field-based surveys often provide new research direction. For example, we are very surprised that the local oyster seeds did not develop abnormal enlargement ovary diseases, although they are maintained in the same fields with contaminated oysters. But that result may suggest the presence of intermediate host or vector organisms of the parasite. So I'm very happy to find the way to do for more. And also, as for franciscellosis of scallops, we still do not know the association between density and the impact of the disease. But the understanding of the things in future, probably we can get more deep insights and deep understanding about the physiological things of scallops. And those results may take us to further countermeasures. And we are very interested in the resistance to the virus in older and larger abalone. Similar things are reported in the Australian herpes viruses. The older oyster shows resistance to the OSHV1. It has been known, but the reason is not clearly understood. So if we can understand why the older or the larger abalone can get a resistance to the viral infection, probably those acknowledgement may provide us a lot of information about the countermeasure against viral diseases. So I think maybe still we need to do a lot of things to do, but I would like to stick on fields and without giving up on finding new countermeasures strategies. So I would like to close my talk with my motto for research. Let's go to the fields. Okay, so thank you very much for your attention.
For sticking on your time. So now we are having 5 minutes for the question. Maybe we can go 2-3 questions to him. So floor is open for discussion now.
Thank you, Dr. Ito, for that wonderful presentation. We don't get any information on molluscan diseases quite often, so this is a very good one. My question is, in your disease investigation on mollusk in Japan, have you encountered any zoonotic pathogens that can pose problems on humans?
Sorry?
Have you encountered any zoonotic pathogens?
I do not think so, and there is no, maybe, of course, norovirus is very serious problem, but it does not cause any problems for production itself. So maybe there is no like that.
That's good to know.
Thank you very much.
Thank you for a really interesting presentation. The life cycle of, let me get the name of the parasite here, Martelioides,
yeah,
is known?
Not yet. And I think at least one intermediate host, but possibly two. So it's very complicated, probably.
So many years ago, I was interested in the fact that EUS appeared off the south coast of Japan and the east coast of Australia almost simultaneously. And I was wondering how it could happen. And I looked at the ocean currents that go past Japan. And on the South Island, you have an ocean current, but going from west to east. But on Hokkaido, it comes the other way.
Yeah.
And I wonder if this is the reason that the oysters around that island remained free of the disease, because it was the prevalent currents that were pushing maybe a free living stage of the parasite from west to east. So is there evidence that it first appeared in the west and moved slowly eastwards in Japan?
Yeah, thank you very much for the suggestion. Okay, so this is just preliminary results, but we found, we collected a lot, zooplankton, and used them for qPCR for this parasite. And sometimes we got positive for certain specific zooplankton species. And according to some references, that plankton is a warm water species. So maybe that's why the parasite was not spread, so it remained in southern western part for long years. But recently, the water temperature around the Miyagi area increased. So that's why maybe that's one of the reasons that the parasite can settle the Miyagi area a little bit, although that area is a little bit colder.
Yes,
Thank you very much.
Thank you very much. The next speaker is coming online. She joined online. So due to the time limit, we'll stop the discussion now.
Okay, the next speaker will come online. She is already here. Professor Claire Gasson. So she's a professor her work is based in the National History Museum of Paris, France. She's a plant and algal pathologist whose research encompasses the physiology and ecology of many pathogens of freshwater and marine algae in polar temperature and tropical environments. Her research spans the physiology and ecology of host pathogen interactions, as well as the socio-economical conservation and also biosecurity challenges that arise from the rapid development of algal cultivation worldwide. Aside from research, she is very proactive in networking and capacity building worldwide to support the development and resilience of the seaweed aquaculture sector. Please welcome Professor Claire Gasson. Okay.
Hello. Hello, can you hear me? Hello?
Yes, we can hear you.
Okay, thank you very much indeed, and thank you very much for this very nice introduction. So, as the introducer just said, I mean, I work on algal diseases, all aspects of algal diseases, and my talk starts with an apology and with a thank you. So an apology for not being able to join here today as I would have wished. This is a great loss for me to not have been able to be in Chennai and meet you. And I would like to really express my gratitude to the organizers who've made still possible my attendance here today by video conference. So thank you very much for enabling this. I guess I'm trying to show, I need to share my screen, sorry. Yeah. So, yeah, so you're hopefully seeing my slides now, and what I want to tell in this talk, I think I will be very brief, because if you're already attending the DAA12 in Chennai, you probably know already that diseases are very important in aquacultures, and seaweed are no exceptions. So effectively, diseases of seaweeds are a worldwide barrier to production, sustainability of the industry, and I'll go very quickly on that. And then I'll move on very quickly to explain a bit about the efforts that we've been making, as a community towards accelerating the description and characterization of seaweed pathogens, and how, we've been trying to go a bit beyond that, from just a pathogen discovery and description to actually setting up a biosecurity framework for seaweed aquaculture. And then I'll finish my talk by presenting a bit of laboratory work that we've been doing, trying to support the design of disease control methods in seaweed, or more generally algal aquaculture. So using probiotics, trying to understand how seaweed's immune system works, and also setting the scene for breeding. So here I would like to acknowledge the work of my colleague Kwon Kim, that's in Korea, and effectively this is really a great epidemiological study that he made about 10 years ago in the 2 biggest Korean farms, and effectively what comes out from these farms that produce pyropia is that if you go in these farms, you will find different pathogens like the green spot disease, the red rot disease, and Opidiopsis disease. I could go into the details of the ecology of these diseases, but what matters to me now is that if you look at this graph, at any point of time, you will have 20 to 30% of the potential production typically lost to one or disease or the other. And what they also showed in this paper back in 2014 was that if you added the cost to the yield plus the manpower that it took to try and rescue the production, this typically amounted to 50% of the running cost of the farms. And then what was very interesting to me at the time, and is also very well known in mollusk and shrimp aquaculture, is that pathogens actually are being discovered as the industry grows, and that was shown here by the fact that Opidiopsis and this agent for the green spot diseases have been described after this epidemiological study was published. And one of the biggest hitter in the Korean industry here in yellow is this Opidiopsis pathogen. So this is where we were 10 years ago with what was known molecularly on Opidiopsis pathogens. So you had in GenBank 3 sequences that were known from Asia, so Japan, Korea, and China. And then you had another 2 phycologists had produced a sequence here in Madagascar and here in the Adriatic Sea. And they were all pathogens of red algae. So with that in mind, and with our interest in Scotland, we started with my postdoc, Yassine Badis at the time, to look at potential pathogens of red algae in Scotland, where there is huge interest to cultivate Porphyra, but also Palmaria palmata. And sure enough, we started finding these pathogens, and we started realizing that there were species of red algal pathogens that were waiting to be described. And then what Yassine also did was that he used the sequences of the pathogens that we knew on red seaweeds to actually query the publicly available environmental DNA data sets that were available at the time. So we downloaded all this DNA data that was available from GenBank and looked for DNA fingerprints that were closely related to our pathogens of seaweeds, the one that were known, and essentially found all these sequences in green and blue, and essentially revealed that there were probably 20 plus different species closely related to the one we knew already, waiting to be described. And then what he also did was to map back these sequences using the GPS coordinate of where they had been found. And, starting from these red stars, which show the places where this pathogen had been found and described, it was basically able to show that you could find either this species or closely related one pretty much in every environment, every coastal environment that had ever been sampled at the time, which led us to conclude that effectively these Opidiopsis pathogens of red seaweeds, even though they were not necessarily described already, were potentially a global threat to red algal cultivation. And then something else that we started to look was at the possibility to transfer pathogens from one place to the other. And this really echoes the previous talk by Professor Ito about the movement of pathogens. So basically what we did was to take the pathogens we found in Scotland and try to infect seaweeds cultivated in Korea. And we took pathogens from Korea and tried to infect seaweeds from Scotland. And of course, it worked. And these are these infections we're showing here. And basically, we showed that non-native Olpidiopsis pathogens could infect both wild and cultivated red algae. And then at the time, what we were seeing was that there was an accumulation of pathogens of algae that were being described, but contrary to the awareness that is already present in the mollusk, in the shrimp and the fish industry, there was literally no awareness amongst the seaweed farmers that we were talking to about the risk of moving pathogens. And there was no quantification of the risk, not least because nobody knew what the pathogens were. And then when we looked at the biosecurity policies, either nationally, regionally, or at supranational level, there was literally mostly no national framework and definitely no international frameworks pertaining to the movement of seaweed and potential pathogens. And that's where we started to work quite a lot, especially with the first global seaweeds projects, which are still pursued. And then one initiative that we tried to lift off the ground was actually to try and accelerate pathogen discovery, saying it's everybody's problem. So it's not a problem where a competition is going to be useful and we all need to collaborate. So we developed this platform, which is now defunct due to lack of funding, but which I'm hoping to resurrect in a way, whereby we were inviting people to effectively contribute photos or samples of diseased algae from wherever it came in the world. If you're interested, we describe this in this 2022 paper by Martina Strittmatter et al. And then the idea was also to be able to use the contributions, the disease reports from farmers that we could even anonymize to preserve commercial sensitivities, but effectively saying, like, we would like to know about the diseases, and if you send us a sample, we will try to return to you a diagnostic for free. And if it is a new pathogen that nobody else knows, then we'll start working on it and try to accelerate the description of the diversity, but also the ecology of these pathogens, and really focus on our efforts on the ones that are causing havoc in the farms. So one outcome of this has been really strongly led by Pedro, who's now a lecturer in Chile. Has been to produce a first version ever of an atlas of the pathogens of algae in Chile, and effectively has found some virus. Well, he's compiled all the records of viruses, bacteria, fungi. We call them pseudofungi, but that would be all my seeds, phytomyxids, all sorts of protists, algal endophytes, parasites. And fundamentally, what it shows is that there are very few records overall, mostly biased towards Chile. But also, what is fairly interesting is that we have records on non-commercial seaweeds, but a lot on commercial seaweeds as well. Brown algae, red algae being affected, and most records in Chile, as I was saying. But the interesting trend as well is that you have here the cumulative number of records, and what you see is that half of the records have been done in the last 25 years as opposed to 1 century. So there's definitely an acceleration of the reporting for diseases, which is directly linked to the expansion of aquaculture and harvesting. So at that point, and I think we are very much lagging behind in the seaweed aquaculture compared to other aquaculture industry, we're really limited by our ability to identify the pathogens. And as Professor Ito was saying, we also are very limited in our understanding of their ecology, which is key information that we would need to then be able to assess the risk to aquaculture activities, but also the risk that aquaculture could pose to conservation of natural stocks, for example, due to movement of pathogens. So just to show you a bit how far we've gone in the last 10 years and how we're still very much working. This is here an example of the diversity of one type of pathogens that are called the oomycetes. So there are different groups of oomycetes. Some of them are well known as plant pathogens, like potato blight causing the grape mildew. And some of you will know very well some oomycete pathogens of fish or crayfish, like Saprolinea or Aphanomyces astaci, that is on crustaceans. And this is the view that we had 10 years ago, where fundamentally there was like in this handbook of protists, there was half a page literally on this group that is called the Alpidopsidaceae, that includes this pathogens of red algae I was mentioning earlier. And since then, basically the amount of described species, the knowledge about their diversity and molecular phylogeny has exploded, to the point that we have now 4 different groups of red algal pathogens, so Alpidiopsis-like pathogens of red algae described. You also have like 2 different groups of brown algal pathogens, as well as microalgal pathogens, including diatoms and dinoflagellates here, and also I forgot 2 green algal ones. So this is really exploding knowledge taxonomically, and the ecology of them is also of great interest to us. I would like to mention here again some work from Pedro in Chile, who has been looking at the co-infection of Porphyra, so this Porphyra here, which is the bladed red seaweed, as well as Bengia, which co-occurs on the coast. It's a filamentous red algae that is not of commercial interest compared to Porphyra. But the point here is that Pedro, looking for infection, has been finding in Chile for the first time ever in South America this Porphyra, which is known from Japan and is causing problems in the Japanese Porphyra and Pyropia industry, and which we had found in Europe as well with VC. So now we know that Alpidiopsis porphyrae occurs in Chile and potentially can infect Porphyra if you're trying to grow it. But what is also interesting was that he's been doing year-long surveys, and I find some seasonality in the occurrence of this pathogen, with some peaks of prevalence can be close to 100%. And our previous speaker was talking about intermediate host, etc. And well, that's pretty much the case, because Bengia can occur in this area and also be infected. And we also have some patterns where, if you sample the same locality, so here on the same days, you can find infected Bengia and Porphyra, but you can also find infected Porphyra and almost immune Bengia, by which at this moment, all we understand is that there are contrasting patterns of infections of possible wild host and formed species that we simply do not understand, but would potentially be critical for disease management. So moving on to the ecology and to more perhaps the policy making, I should really credit the work of other people. So the first thing that we did was trying to look at how we could improve the biosecurity, our own hatchery, and also try to, explain and make recommendations for best practices to seaweed farmers. And here I need to credit Phil Kerrison, who was working with me and the Scottish Association for Marine Science for running a sort of biosecurity upgrade of our hatchery, which, as you see, is a very modest one. And the general idea was to make sure that we were not getting pathogens from outside, but also make sure that we will not throw anything, discharge any pathogen, if we had any in our hatchery, outside to the waters of the bay that we're having. I won't get into the details of the disinfection procedures, but they're all publicly available online at this URL. We were also trying to address the lack of awareness about diseases amongst farmers, policymakers, etc., by producing resources. I've just tried to highlight some about Asia that we did, and also trying to credit my collaborators on this, especially at the University of Malaya and the Natural History Museum of London. So that was basically in this Asia Diagnostic Guide to Aquatic Diseases, that was the first time ever that seaweeds were included in the manual on disease diagnostic in aquaculture. Doesn't look like much, but this was really us trying to say, hello, seaweed pathogens, we exist. And then there have been a number of other initiatives here led by my Malaysian colleagues, Peking Lim, Professor Peking Lim in particular, about producing resources for farmers, including in local languages. So trying to make that knowledge accessible to people who do not use English as a language. I've also been involved for the last 10 years, placed in training schools, trying to teach students of all horizons about diagnosing seaweed pathogens. And if you're interested, this would be the latest one that was funded by the European Union. And I would like to credit very much the work of my colleague Les Coutier-Cook at the Scottish Association for Marine Science to try and really assess and improve the national and global biosecurity framework for seaweeds and also the disease management practices. So there's a number of work that she really led on this, about trying to understand the practices and to develop risk assessment tools to predict disease outbreaks. She's been working with Indonesian colleagues, Tanzanian colleagues, and the Philippines as well, working at the global on the global stage, especially through the United Nations, FAO, and Safe Seaweed coalitions. And fundamentally, she's very proactive in this work, now working with Thailand, for example. So a great deal of work that she's been carrying, and I think is really outstanding. So if I just finish my talk about some more theoretical and upstream work that we're doing in the lab about disease control methods, I'm just going to present a very simple scheme of what we've been trying to assess. Fundamentally, if you're thinking of a seaweed that you're trying to cultivate and a pathogen, the most obvious solution is you'd like to hit the pathogen with something toxic, but pretty much nobody wants to do that, because whatever is toxic to this pathogen might be toxic to a whole lot of other things in the sea. And this is probably going to be a very destructive control method. So we have been very much looking at efforts of making the alga stronger, and that would be using biotics or enhancing the defenses. There are quite a number of articles on this. We did a bit of this, but that's not really the most of our work. A lot of our work is trying to set the tools that would be needed for breeding disease-resistant algae, and also a lot working on the microbes that are naturally associated to the seaweeds, and trying to understand if we could use naturally occurring microbes associated to the seaweeds, either to enhance the seaweed defenses, so make the alga stronger, or directly to compete or hit or destroy the pathogen. So looking at how we could use the microbes, this is work that's been done with a very dear colleague of the museum here in Paris, Soisé Prado. We started with isolating fungi and bacteria associated with the kelps, and I think that's this paper here. And then we started to look whether some of them would have an activity against these pathogens. And we started identifying molecules that were produced by these microbes, which could have direct antimicrobial activity. So that's the case here for this pyranocin molecule here. And what we also found was a group of fungal produced molecules that were having an activity against the bacterial quorum sensing signaling. So these are heterocyclic molecules here. And the general idea is that this fungi could then interfere with the signaling of bacteria, and some of these bacteria being pathogens, we have at least a theoretical possibility to use these fungi or the molecules that they produce to try to improve the resistance of seaweeds against bacterial diseases. To be honest, that's pretty much where we are at the moment, and we do not have solutions. We're just trying to explore what would be possible. Another model that we've been working on very recently is actually on a freshwater microalga, Haematococcus, that is grown for the pigments. And this is work here done with my PhD student at the time, Jeanne Miebach, where basically we realized using actinic cultures that bacteria naturally associated to Haematococcus are necessary for the infection of Haematococcus by a pathogen fungus called Paraphyseterma psittaciborense. So that sounds a bit counterintuitive, but basically we showed that bacteria are critically involved in the success of the fungal pathogen. And then we felt, well, there must be some bacteria that also are present around Haematococcus and can help modulate and hopefully decrease the infection. And we did a lot of work on this. And what we have at the moment is we haven't found bacteria that produce the disease just yet. But what we see is that we have a reduced cohort of bacteria. It is structured, it's not random. And basically, this has a lot of implications in terms of if we want to modulate the bacteria, the bacterial microbiota around the alga, all this data that we've acquired is going to be absolutely critical if we want to have successful pathogen biocontrol. So very quickly, in the time that I've got left, I would like to move on to the work on the immune systems of algae. And here I'm going to present some work that we've done on brown algae, including kelps, but also model brown algae like Ectocarpus. And essentially, this is a very complex story that I'm trying to sum up in one slide, whereby we showed that across all brown algae that we've tested, so the whole phylum, using 3 different pathogens, so 2 omycete, 1 phytomyxid, we basically have a two-layered immune system. The first one is innate, is local, and this is really the resistance to the pathogen is mediated by the local programmed cell death of this pathogen. And afterwards, when the epidemic outbreak continues, we have the induction of autophagic responses, both in the host and the pathogen, and that also contribute ultimately to the host defenses against the pathogen. I'm sorry, I don't have time to get into the details, but this is really, there are 2 papers already out in New Phytologist iScience that I could refer you to that. Using this knowledge about the immune systems, we've identified markers of programmed cell death and autophagy, which we can then follow as markers of the onset of resistance. And we've started to develop non-invasive methods for the quick bioassays to test for resistance, so that we could screen basically lots of different seaweeds as to whether they were resistant or susceptible to disease. So a first method that we've used, and I think that would be usable also for organisms outside seaweeds, mollusk, shrimps, etc., is nephelometry as a non-invasive tool to measure the growth of our seaweed, in our case. And then we can combine nephelometry with fluorescence, and we can do measure chlorophyll fluorescence. We can use fluorescent assay to mark our pathogen. And we've also been using a lot of different techniques to measure how much the alga is suffering from the disease or how much the pathogen is progressing. And we're getting a number of assays in microwell plates. And one of the latest technique that we're doing as well is high throughput imaging. So illustrated here on Haematococcus infected by this fungal pathogen I was just talking about, Paraphyseterma psittaciborense. And fundamentally, what we're seeing here in our cultures is that the Haematococcus cells do not get equally infected. Some of them have a lot of pathogen spores aggregated, whereas most of them are not infected. So in order to measure that heterogeneity within our cultures, we use a whole slide scanner, then AI-aided image analysis to detect and then quantify the cells, followed by a manual check. And that allows us to quantify our pathogen even at very low prevalence, so at the very beginning of the outbreak. And then we can see this aggregation that was not known for algal pathogens. And then we're thinking that if we can disrupt this aggregation of the pathogen spores on the very few first algal cell get infected, then we would have a way to control the infection outbreaks in a cultivation setting. And this is my very last slide. This is really to flag up an international collaborative effort that we're running thanks to funding by the EU and UK RISE. So that's a project, it's called FAB, whereby with Yassine Badis, Pedro Murua, but also Sigrid Neuhauser and Peter Van Best in the UK, we're effectively having, I would say, a great bunch of PhD students working on different intracellular pathogens of algae. So we have Sarah working on the my seeds of red algae. We have Joan Lewis taking over the immune system, the work on the immune system of the brown algae. We have Leah Teifel working on the phytomyxid, intracellular pathogen of browns. Yassine, Pedro, and I still work on the my seeds. Pathogens of brown algae. And then Peter is picking up some work that on the my seeds pathogens of diatoms. And we're hoping to compare the infection strategies of these different pathogens between each other, and also the defenses of the different algal hosts to really improve our knowledge of these host pathogen interactions across a number of seaweeds and microalgae. And with that, I would like to, thank you again for your attention and to take any question you might have. Thank you very much.
We have time for 1-2 questions from the floor. Are there any questions? Yeah, Melba.
Good afternoon, everyone. Professor Claire, thank you very much for your presentation. It's not a question, but it's really a comment to inform everyone that seaweed is going to be the next superfood in the future. And I am very happy to see, I'm involved with the work of Professor Elizabeth, and the seaweed sector, or the seaweed commodity, is the very first commodity that trying to implement this, what we call as the progressive management pathway for improving aquaculture biosecurity. And you are very right that it's only a matter of time that any food production, as it goes into intensification of production, diversification, movement, 101%, in the absence of any biosecurity measures, it will encounter diseases. Before, we only know of what is called ice disease, and I think until now it has not been fully identified the causative agent. So I encourage the fish health section, especially the new generation in the next DAA, since the approach of DAA is sort of commodity-based or sector-based. You know, we tackle finfish, shrimp. I think that it's now high time to devote more time and encourage researchers to put their efforts in understanding diseases of seaweeds. I'm very happy Ito-san made some presentation on mollusks. So mollusk and seaweed are our lesser brothers in this conversation. So in the future, I hope that we can encourage researchers to focus some of their attention on mollusk and seaweed, and especially Asia. I don't have the statistics. Rohana, you showed something on aquatic plants. And we will encounter so many problems. One, you have mentioned reporting, and I think Ed is here. I don't know if Alicia is here already. This is reporting for seaweed because it's not an animal. It's an aquatic plant. So whether WOA will take it or IPPC or, well, any standard setting body should take up, be prepared for reporting mechanism that will be established with respect to seaweed diseases. Thank you very much. Professor Claire, thank you very much.
Thank you very much for this very nice comment. Very thoughtful.
Oh, we have one more question here.
Thank you very much, Professor Claire. That was a very exciting presentation. Do we get to learn something from the algal blooms that take place in the ocean and the disappearance of the blooms? Are they due to diseases or what really crashes them? Because in the succession of blooms, invariably when they happen, there are reasons that of course ascribe to them and then they disappear. So can we learn some lessons from there in order to talk about what happens inland during culture? Thank you.
So yes, we can, or we hope to do so. And I mean, I should say that in FAB, there is another 5 PhD students who are working actually on the pathogens that are associated to algal bloom. So there's Paul and Deepak are working on the items, but we also have a whole lot of PhD students working on cyanobacterial bloom and how they could potentially be controlled I would say naturally, but also biocontrolled potentially using pathogens. So we are trying to inform the research on seaweed farming and microalgal farming, but what we get in blooms and vice versa. I should say that in terms of using pathogens to control harmful algal blooms, I still think like we lack the understanding of the possible side effects. So because we are just discovering the pathogens as we go and we have very little understanding of their ecology, we have basically no understanding whatsoever about the potential risk to non-target organisms, I would say non-target microalgae that would be key, for example, in the food chain, and that could be the inadvertent targets of biocontrol. So yes, I mean, it's in our mind. We're definitely working on this, but we do not have, I would say, ready-made recipes just yet. If that's your question.
Yeah, but say, for instance, Noctiluca with the pedinomonas in it, you know, these sometimes the nano, forms that are within some of these cyanobacteria, the larger cyanobacteria, see, they also are a very important problem because of all the toxins that they produce.
Absolutely,
right?
Yeah.
So whenever we are talking about diseases in algae, of course, it's very important how to control the disease, but also to see whether if we get some lessons from the natural controls, can they be adopted as a biotech measure? That's what I was
Sure.
I saw one of our students, Deepak Pinto, in the slide that you put up. He spent 2 years with me, a bioinformatician. Yeah. Thank you very much. Yeah.
Okay.
Just a quick one, not a question, but a follow-up on Melba. So is Claire still online?
Yes, is that you?
Okay,
Victor?
yeah, Dr. Claire, thank you very much. And with those roster of students that you flashed, we hope that some of them will be present in the next DAA13. We will announce the venue later, so I don't want to preempt. And also Dr. Ito for molluscan diseases. So we hope to see a few paper presentations on diseases of seaweeds and diseases of mollusks. Thank you.
Thank you very much.
Okay, thank you very much. So I think we already 20 minutes late. So what do we do next? Yeah. So we can stop the session now. Okay. Then I think we should stop the session now and then start it.
Okay, thank you very much for having me.
Excuse me, yeah. Thank you very much.